Good morning, dear students. I, Dr. Mahesh Singh, welcome once again in School of Agriculture. Today we will study about the mango malformation under disease of field and horticultural crops and their management. The objective of the today class is to teach the students about symptoms, etiology, disease cycles, and suitable management practices of the mango malformation for better and assured yield. You can see here the diseased or infected portion of the mango that is malformed by the pathogens, and your objective is to treat this infected parts of the mango to healthy one. This is the healthy one inflorescence of the mango. That's why the learning outcome of this study is to, at the end of the lecture, students will be able to understand about the importance of the diseases, their causal agents, their symptoms, favorable condition at temperature, at what temperature or what humidity condition the disease may spread, the disease cycle, and suitable management practices of the disease. Now, about the mango malformation. As you know, the mango is a king of fruits. The Magnifera indica is also considered as the king of fruit. The mango malformation is a fungal disease. It was reported for the first time from Darbhanga, Bihar in India. In India, it produced nearly about 60% of the world mango, that is about 9.5 million tons. Unfortunately, this crop suffers regularly huge losses due to malformation, a serious threat to mango industry. This disease is one of the most destructive in nature because the economic losses is reported about 5 to 70% or sometimes more than that at severe condition. Mango malformation is the production of thick vegetative shoots at the growing tip and transformation of the floral part into the compact masses of the sterile flower. Two types of the malformation have been observed, that is vegetative malformation and floral malformation. Now we will further study in detail about the vegetative and the floral malformation. Now the causal organism. The causal organism, this is also known as pathogen. Pathogen, earlier it was considered as this is the viral disease, or this may be due to the any insect infection, but disorder also. But now it has been clear that this is due to the fungal pathogen, namely Fusarium moniliforme pathovar subglutinans. The microconidia are one or two celled. It is you can see here the microconidia are shown here in figure. This is the microconidia. It is a septate microconidia in ovals and fusiform shape, and the microconidia are rarely produced. They are 2 to 3 celled and pleasant chlamydospores. You can see here the chlamydospores, and the mycelium is also shown here in figure. And now symptoms. We will see the symptoms of the pathogen uh, and on this disease. There are two main type of the symptoms. You can see here the vegetative malformation and floral malformation. Vegetative malformation, you can see here the vegetative plants at nursery level, and the floral malformation at the time of the flowering or after that. First one is vegetative malformation, and simple vegetative malformation, also known as in short VM. is more commonly found in young seedling. Basically, it is found in nursery condition or at the branching out tip also. The seedling produces small shootlets bearing small scaly leaves. You can see here the very small scaly leaves listed over here. You can see here the small scaly leaves instead of this healthy mango leaves. Like that's why this looks like a bushes or bunchy appearance uh, at the apex. The plant or seedlings remain stunted in growth. The development of the seedling have restricted due to the cause of this pathogen at normal growth. Next one is a floral malformation. In short, it is also known as FM, floral malformation. And at floral malformation, it considering the severity of the malformation may vary on the same shoot from light to medium or heavy malformation of panicles. Heavily malformed panicles are compact and overcrowded due to the larger flowers. They continue to grow and remain as black masses of dry tissue during the summer, but some of them continue to grow till the next season. You can see here the compaction of the flower malformed flower of the inflorescence. The panicle get compact at the summer and it causes the heavily mosses of the conidia or the pathogen. Now, disease cycle. The how the pathogens can continue or their life cycle from one season to another season, or how they spread from one place to another place or one plant to another plant. Malformed inflorescence and vegetative growth serves as a source of the pathogen of the conidia, which reach infection site by at least three different means, that is wind, insect, and via host materials, infected host material that falls into the funnel-like structure of the apical buds. You had already seen that the apical buds are just like a bushy appearance and funnel-like appearance, and the conidia may fall down after the traveling by the winds and get established there and cause the disease over there. Now, fungus grow rapidly there when the favorable conditions are suitable there. Weather and climatic condition at high humidity, moisture are there, and at that temperature and favorable conditions, conidia may rapidly grow there and causes the infection. The presence of Assyria magniferi, the presence of Assyria magnif it is a like a mite. It is considered as a mango bud mite at apical portion. The frequency and severity causes by the pathogens. And after the host penetration, the pathogen colonize buds but not subtending tissues, and whether infected birds subsequently developed symptoms of mango malform disease apparently depend on the extent to which they are colonized. It appears that minimum threshold of infection by colonizing mycelium uh, is required of vegetative and floral symptoms to appear. Shoots that were only slightly colonized were never symptomatic. Means the pathogens basically transmit through the wind, through infected tools or materials, and through rain splashes or rain droplets. Now you can see here the disease cycle also represented by Graphical portion, apical bud colonize with Fusarium magniferi and Acaria magniferi. This causes the infection start due to this the symptomless branches colonize with the Fusarium magniferi at you can see the some branches may be asymptomatic condition, and the pathogens survive over there, and uh, the after the maturity of the pathogens, they spread through air, and the spores you can see here through air current. They disperse from one place to another place, and the direct there are two portion over there. First one is direct bud infection, and second one is through mites. This mites falls on the healthy leaves and cause the infection, and direct infection may also cause the conidia falls down on the infected healthy portion of the apical portion of the plants, and they cause the infection over there. And after that, these plants get um, this colonized apical bud with malformed portions. The malformed portion also get vegetative growths of the plants, and this the vegetative growths after maturity, they elaborate, they burst the conidia, and the conidia also further disperse over there. The dry masses also malformed plant, they fall on the plant debris directly and cause the disease over there. Now, the disease management. Diseased plants should be destroyed at initial level. At the initial level, when you find the initial symptoms in your orchard, just you have to identify this type of the pathogen symptoms and just cut along with some 20 to 25 centimeter with healthy portion and destroy them, or at nursery level just destroy the whole plants of the infected. Use disease-free planting materials. You have to use disease-free planting material for the orchard development. Scion and its stalks from infected trees should not be used when you have to use the uh, nursery growing, that is cyan and stock. It should be take from the healthy tree or healthy plant parts. As soon as the disease appears, the affected terminals along with the basal 15 to 20 centimeter, as I earlier told you, are more than 20 to 25 centimeter, healthy portion should be removed and prune out and burn that portion. Mango varieties like Bhaduran, Elachi, Lal Sundari, H811, that is a cross of Amrapali and Lal Sundari, or Pitambar, that is a cross of Amrapali and Lal Sundari. Uh, this is also resistant variety, and this is the Bhadya, Dula, and Malda also, and this is Langra, and it is also resistant against the mango malformation. We have been identify the resistant sources to develop the variety resistant to mango malformation, and we can chemically also treat this malformed pathogens against pathogens. First, we can spray the 100 to 200 ppm of NAA, that is naphthalic acetic acid, during the month of October, and we have to spray or apply the Planofix at the rate of 200 ppm during the first week of October, and followed by the de-blossoming at bud burst stage is recommended. A single foliar application of 1000 ppm cobalt sulfate prior to flower bud differentiation successfully reduce the floral malformation. Means we have properly managed the disease by applying the sequence wise of this disease management. Now, practices question, dear students. There are some practices questions, and you can practice of these questions in your notebook. Uh, First one is, what is the malformation disease? I already discussed in detail. You have to answer the malformation, about malformation disease, their symptoms, their causal agent in detail. Next one is, how do you control the mango malformation disease? I already discussed you the management portion of the disease, and you have to answer in your details about the mango malformation disease of the control management practices. Next one is, write the disease cycles of the mango malformation disease. You have to write the disease cycles only over here, and you can draw the picture also. Next one is, write the causal agent and symptoms of the mango malformation disease. You can just write the causal agent, that is pathogens, name of the pathogens, and and the symptoms. Symptoms, there are two types of symptoms. I already told you that you have to write over there. And next one is, what are the cultural and biological management practices? There are some cultural practices and biological management practices also over there of this disease. You have to write in your notebook. Now, these are the some references you have to study in detail over there, and you can click on this link page, or this is the some textbook is listed over here, and you can study in details. Thank you very much, dear students.